start.
All right, very good morning, everybody. All the students and participants of the MOOC course Interactomics who are joining us today. It is a long course, 12 weeks. I hope you have had a good opportunity to go through multiple technologies and learn new ways of understanding the protein-protein interaction, other biomolecular interaction. Today, we thought not to just give you the lecture, but rather give you some lab demonstrations to give you the first-hand feel of how some of these experiments, which we have been talking in theory, how they can be performed in the actual lab settings. So the theme for today's live session and the lab demonstrations is about protein microarrays and label-free biosensors for various proteomic applications. You have been interacting with Dr. Argya. He is the main TA for this course, and I'm Sanjeeva Srivastava, the instructor for this course. So as you have studied already in the course that we have various type of protein microarray platform. And right now when we are talking about protein microarray, unlike the DNA oligo-based microarrays, what we are talking is some very difficult class of molecule to really print on the chip. Because protein, as you know, it's very difficult to purify the protein. It's very difficult to express the protein. It's very difficult to print the protein on the chip. So therefore, protein microarrays are not as simple. And, in the analogy, it's not like the DNA microarrays. It's very difficult. And therefore, different type of platforms have emerged, which are aiming to investigate the proteins in different type of forms. For instance, you can take if you're very lucky and you have, let's say, you're looking at yeast proteome, you have purified 5,500 or so proteins. You're looking at a bacterial proteome and you have purified maybe all the 1,000 protein of that bacteria. Those are still, I think, somewhat doable. But if you think about, looking at the human, for example, human proteome, and we talk about 20,300 genes and those protein, it's really very, challenging task, right? Quite mammoth task. So ideally, when I say protein microarray, aim or our assumption is we are looking at purified proteins which are printed on the chips. And those proteins, then we are going to have miniaturized high density platform. And then we are going to utilize different properties, looking at sometimes the purified protein to test the interaction, sometimes the patient samples to look at the autoantibody response. So various type of assays could be performed. And often we also look at the protein small molecular interactions or look at the post-translational modifications. We can also look at different type of enzyme substrate assays. So many type of things can be planned. But idea is, can we have those many purified protein? Let's say if you don't have that many purified protein, then alternative approaches have also been utilized. For example, you have studied NAPA, nucleic acid programmable protein array. And likewise, there are many other cell-free expression platforms which we have been talking, right? Like the PISA and MIST, multiple spotting technology, protein in situ arrays. Many technologies are available, which are also utilized to take the DNA and print the DNA on the chip and then express that in the in vitro transcription translation method and make the protein on the chip itself. So based on your content, you can try to create the protein microarrays, which could be utilized for analytical application, which could also be utilized for looking at the function of the proteins. And therefore, we can term them as the analytical protein microarrays or functional protein microarrays. Additionally, a lot of clinical application and clinicians, they look for some abundant biomarkers. For example, if the elderly men, they are going for test for the prostate cancer, a small tissue will be printed, let's say from 1,000 patient on the small chip. And all they are doing is they're looking at anti-PSA antibody, which is going to look at the abundance of that antibody in every patient sample. So now what we are talking is we are looking at how the different patients are expressing the level of PSA protein. So this is, reverse phase protein microarrays when we are actually printing the live cell or the tissue, and then we are using the antibody-based approach to look at the specific type of abundance of molecule. So all you can see here, or you might have just got refreshed with the flavor that, protein microarrays can be prepared in different manner based on different type of applications. So for any type of protein microarrays or peptide microarrays, we start with blocking the chip because in addition to the proteins that are printed on the chip, there's a lot of empty surface. And that empty surface we want to first block with the BSA in the milk, or we also sometimes look at the blocking buffer, super block, many type of commercial solutions available as well. Then often we are looking at the humoral immune response or autoantibody response. So we look at if a patient is suffering from a disease, are there some antibodies raised against that disease? And that's primarily very abundant in the autoimmune disorders. But even in different type of cancer, people have hypothesis that there might be antibodies which are already made much ahead of time, which could be utilized and screened on the chip. Then we are looking at further, since we are looking at autoantibody response, we can incubate that with the anti-human IgG or anti-human IgM, and then look at those abundance and scan the slides and do the data analysis. So this kind of a typical protein microarray workflow, which I have showed you on the left side. On the right side, I'm just giving you one example of, we have been investigating different type of diseases, including some of the brain tumors. For example, gliomas, meningiomas, various other CNS tumors. Arya will talk to you shortly about pituitary adenomas. So idea is like, on this is, on the B panel, you can see high density human proteome array, which contains almost 19,500 protein. Every protein is printed in duplicate along with many control spots and the empty spots. And then we are probing the patient sample. Then we look at the response of an GST to look at the protein expression, or we also look at the IgG response to look at the immune response. So this is something which is an interesting approach of screening lot of disease-specific antibodies using the high-density protein microarrays. I'll leave the application part with Arya. He will talk to you about that. But I'll just also mention to you, like, often we, rather than looking at the entire proteome, we can also look at the peptides, because if you think about, we have been also talking about mass spectrometers. Mass spectrometry gives us idea for which are the peptide sequence which are very abundant in a given disease. So now let's say for a given protein, you have identified 5 peptides which are very interesting. Their expression level is very high, always in the disease condition as compared to the control. So now rather than making the full length protein, why not, you synthesize only those peptides, synthetic peptide, and print them on the chip. So you don't have to go through the protein purification work. So peptide microarrays have also come as a strong alternative to the protein microarrays, and that's much more relevant, especially if you want to take your discovery forward from the mass spectrometry-based approaches to the peptide-based microarray. During COVID time, we were looking at SARS-CoV-2 virus and its proteome analysis. In collaboration with a company from Germany, we got this chip of SARS-CoV-2 peptide proteome array. Idea is like all the proteins of this particular virus, 15 amino acid long peptides were synthesized in the tiling manner, and all those peptides are printed on the chip, and that chip was now screened for the COVID-19 patient samples. Some of these patients were mild, some of these were severe form, and we're looking at what kind of humoral immune response these patients illustrate. So I'm now going to give you a short video demonstration about how peptide microarrays we have used during the COVID time, but for that matter, this can be also utilized for any other applications as well. So I'll now play this short video, and then we'll go to the lab.
Today, we will be demonstrating peptide microarray technology. The first step involves reagent preparation, namely washing buffer, staining buffer, and Rockland blocking buffer. After preparing the reagent, we move on to incubation tray assembly. Unscrew the incubation tray by slowly turning the screws diagonally. We will be using 2 blank glass slides as balancers, since today we will be using only 1 microarray chip. This is the pepperprint microarray chip. Slowly take out the chip using plastic forceps and place it on the incubation tray. Align the chips properly onto the tray. Now, allow the gasket to be placed over the slides. Remember to keep the glossy side of the gasket on the downward side. Align the silicone gasket over the slide and tightly seal the incubation chamber using the screw. Remember, tighten the screws only diagonally. This will ensure that the slides do not break. Next, add 2 ml of staining buffer onto the chips. This step is called equilibration of peptide microarray chip. Please note that we are not touching the microarray chip at any point. Now allow the equilibration process to happen for 15 minutes. Now aspirate and remove the buffer and add 2 ml of blocking buffer. Slowly add the buffer onto the slides. Allow for 30 minutes of incubation. The next step is sample incubation. To the staining buffer, we will add patient plasma at a dilution of 1:200. This is the patient plasma. Now slowly overlay the diluted plasma over the slide and incubate overnight. This will be done in a cold room at 4 degrees Celsius at 140 RPM on an orbital shaker. On the next day, remove the incubation tray from 4 degrees Celsius and aspirate the solution. Now add washing buffer thrice onto the chip. This step is done 3 times in the dark. Again, completely aspirate the washing buffer and add secondary antibody. Prepare of dilution of secondary antibody 1:2000 and then slowly overlay the secondary antibody on the microarray chip. For 45 minutes, allow the incubation to happen at 140 RPM at room temperature. Make sure that you do not expose the slides to light at any given moment. Now remove the buffer and wash the slide thrice using washing buffer. Now gently take out the slide and put it in dipping buffer. This is done to wash away all the salts that may be over the peptide chip. Use a pressurized stream of nitrogen to completely dry the slides. Finally, we will scan the slide. This is the microarray scanner, GenePix 4000B. We are using manufacturer recommended settings for scanning the slide.
All right, so you got some idea that how a peptide microarray, and in a similar manner, even protein microarray experiment can be performed. So now we'll go to the lab and walk you through different steps involved in protein microarray. I request TAs Vaishali and Dr. Argya to now show you the live chips as well as the scanning parameters from the lab.
So hello everyone, my name is Vaishali. And today we are going to understand the basic concepts of microarray, and we will move specifically to protein microarrays. So if you come, so this is the GenePix microarray protein microarray scanner. So basically what microarray is a high throughput platform where we can analyze thousands of biological interactions on a single solid surface, typically a glass side, which looks like this. The glass side. And so there are many types of microarrays that we can study based on what biological interaction we want to study.
So basically, you can see a high density microarray slide in which around 18,000 proteins have been spotted in replicate. So these are the HuProt chips which have been manufactured by CD John Hopkins.
So there are many applications that we can do using microarray. We can study DNA hybridization. We can study protein interaction. We can study antigen antibody interaction and many more. So
you can place.
How we insert a chip in the microarray scanner. We put the slide in it and we run the slide, and this is how the image of the slide comes out. So we can see there are many fluorescent spots if I enlarge the image. So you can see that these spots are the fluorescent spots that are identified from this detector. So basically there are 4 key steps of the workflow. First is that we do the protein immobilization, specifically for protein microarray. So the key proteins are immobilized on the glass surface, and then we incubate our biological samples, be it plasma, be it serum. So they have primary antibodies which bind to these proteins that are immobilized on the surface. Now we add secondary antibodies which are fluorescently tagged. These secondary antibodies bind to the primary antibodies, and after detection, it gives the image like this. So these are the spots or called features. They are termed as features. So any slide is printed in such a fashion that they have these blocks which have similar proteins in each block. So meaning that we can analyze about 14 or 16 samples at the same slide, same time. That is what it means when we say it is high throughput and it has high reliability. So now if we want to check the quality check of the block, so the proteins are immobilized in such a manner that the first 2 blocks, or essentially more than 2 rows or columns of this particular block, has same proteins. So if we can see the first row and the second row of this particular block, they have same proteins printed on this. So we can analyze from the intensity values and the spot intensity. They should be same. If they have different signals or different intensity values, then we have to discard that the quality check of that particular block or slide is not good. So we can do QC or quality check of the data itself.
So I will open up one new slides giving the live demonstration of it. So giving the live demonstration of it. So this you can find a CDI HuProt chip. This is how the arrays look like. So So this is how the arrays look like after scanning. As professor indicated, there are the quality control proteins, which are the green fluorescent proteins. We measure the quality control proteins, green fluorescent proteins. You have to optimize all these protocols. Like you see each protein spots, there is oversaturation. You have to find the optimal wavelength at which the spots are clearly distinguished, and you can find like all these protein spots are represented. These are the GST spots, which indicate like from the increasing concentration of 10x, 20x, 50x. You can use this for the quality control for the green channel. Now I will open up one real cancer based applications. What you can see, uh, so this is the red channel what I am opening, in which rhodamine labeled antibodies are there, and these fine spots what you are seeing are the labeled antibodies. So these are the GST, and whatever like in this area you are finding the pink spots are which are coming are the your auto antibodies which have been screened. So you can optimize the protocol based on your requirement. The auto antibody detection, you can use the chips which are labeled with rhodamine. Various other QC proteins are also there, but we generally have used the rhodamines. So this is one of the unique quality. As you can see, there are all the lasers which is inside. You can scan the chip from here, and once the scanning is done with the help of the GenePix Pro software, you can detect the results. So this is the demonstration of the microarray. So back to you, Prof.
Thank you, Arya. So I guess the students, you've been already studying this protein microarray and peptide microarray in detail. So we thought not to spend too much time on it today, and rather give you a little more flavor of other applications as well. So given that what we have seen, that protein and peptide microarray, they require some sort of tagging, some sort of labeling, and often we use the fluorescent-based tags, right? These are very commonly utilized reagents which every lab has, and most of the time scanners are available which can scan the fluorescent reagents. So therefore, we see them, being used very commonly, whether some of the Western blot, different type of other fluorescent-based scanning which we see, and microarrays. These are very commonly utilized. But if you think about, we are looking at biological function, and if we are adding some tag or we are adding some reagent along with the protein, it's going to mask the functional property. So therefore, adding the tags are always not very good way of utilizing the protein-protein interaction or protein biomolecular interaction properties. Also, protein microarrays are very laborious experiment. Forget that what time it takes to make the arrays, and which is very cumbersome. But even beyond that, once you have the chip and you are doing the experiment, it takes good 1.5 to 2 days time, starting from blocking to various type of incubation, and then only we are able to get any kind of readout, any kind of signal, as you have seen in the scanner, the slides, that we could get some images from that. Now, after looking at the scanned images, then we are only able to understand if the experiment has worked out or not worked out, right? So, for example, there is too much background, or there is a good signal, but there is a lot of spillover. The concentration is too low. Many things we only learn after scanning the slide. So it means we are looking at endpoint measurements, which is one of the very big drawback of utilizing these label-based platform. Like you're doing the experiment in a blinded manner for almost 2 days, and at the end you are very disappointed experiment did not work out. So alternative approaches are label-free technologies, where we are looking at the inherent properties of two biomolecules. If two molecules are interacting, there will be some change, there will be some mass change, some dielectric property change, interference change. There will be some change happening when two molecules are colliding. Can we not label them, but rather look at what are the changes happening on these proteins based on the other molecular changes happening with the inherent properties? So now different type of label-free approaches has come forward, which are utilizing this approach to look at the measurements in the label-free manner. Additionally, these technologies also gives you the readout in a very live manner, like you're looking at the biomolecular interaction happening, and you start measuring them from the beginning of the experiment. If you see there is no interaction happening, then you are going to try and make some changes and say, okay, can I increase the temperature? Can I increase the concentration? Can I try some other dilution? So many things you can change in the live experiments. So therefore, you are looking at the real-time measurements and optimization and troubleshooting in real-time manner. That gives you a lot more edge in doing the label-free biosensors-based platform. So while you have probably already studied this in the my lectures, just refreshing you, one of the key technology in the label-free biosensor, which has been the gold standard in the field, is known as SPR, surface plasmon resonance. Here we are looking at the molecules are immobilized on a gold slide, a light source and the prism is there. You are looking at the refractive index of medium getting changed in response to the binding of the molecule. When the light is excited, the surface plasmons are generated. And when two molecules are binding, an antigen antibody binding or two proteins are binding, we are looking at the SPR reflectivity change, and that we are measuring across the runtime to obtain the a chromatogram, which is now known as here a sensorgram, SPR sensorgram, which you can see in this kind of a graph, where we can see initially there is a baseline, there is no interaction happening. As you are adding the query molecule, slowly you can see the on rate or the some binding is happening, which is K small a, and over the time gets saturated. You keep adding the buffer, and that actually comes to the dissociation rate, and eventually you want to utilize the gold chip again for your experiment, so you can now do the regeneration with the mild acids. So this is kind of just refreshing you the concept. But now idea is, can we show you this live in the lab? So I request now Rucha Gadre at IT Bombay, who is the leading the SPR facility, and she's going to give a demonstration along with Argya to talk to you about BIA core technology, which is one of the key technology to utilize for the SPR-based protein interaction. So over to you, Rucha. I am making you on the full screen now.
Hello everyone, this is Biako T200 from Scientific. We use Biako T200 to study biomolecular interaction. So we study different kinds of biomolecular interaction, like protein, small molecule, protein with DNA, drug molecule, all sort of biomolecular interaction we study. We find KD on basis of change of rate of association and the dissociation. So what happens? We immobilize one of the interacting partner and we move the other partner. And as the binding occurs, we see there is a change in, we see the changes. What kind of changes we see? So for example, this is the principle. We immobilize one of the interacting partner on the chip. These are the chips. So chip has gold surface, and we immobilize interacting partner on basis of some different chemistries. So when there is a light hit on chip, there is a total internal reflection. And as something bound on the chip, there is a change in this angle. The detector will detect this change in angle, that is called as response unit. Response, and we get the graph in terms of sensorgram. This is how the sensorgram typically looks. Okay. So now this is the instrument. This is where we insert the chip. This is where we pick the samples. This is where buffer is kept. After inserting the chip, we perform the assay. And this is how this sensorgram looks. So this is association and this is dissociation. And so you can typically see. So this is how there is a change in rate of association constant and rate of dissociation constant. This is how typically the data looks. I will show you different data so that we can see how this change in association, dissociation look for the different combinations. So you can see this is one protein-protein interaction. You can see this association and association. You can see the difference of association and the dissociation. This is how we study different proteins. Now you can see in this. Now see, for example, this it is not dissociating. It is showing that it is a very strong binder. So this is how we see the different data. It is associating and dissociating. So this will give you
KD, that is equilibrium dissociation constant. And
So this is one of the powerful and robust tool. This is the Bioco T200 SPR, surface plasmon resonance, which can be used for the detection of the KD and Kon and where the interactors can be bind. You have seen the gold plated chip where the interaction happens, and we have also explained the principle of how the SPR works. Back to you, Prof.
All right. So again, like, SPR is used for many industrial applications. People, a lot of industries, they look at characterization of their small molecule. They look at characterization of their antibodies, monoclonal antibody, which clones are having the good affinity. Many of these things are very industry standard to look at surface plasmon resonance-based binding to determine their KD values. Now think about, like, different drugs are being taken for different type of activities. Like sometimes you need a drug which you want to work immediately. So you are looking at very strong, a quick on rate. While some drugs you really want to see them, getting released very slowly, and they should work for a longer time. So you are looking at, very slow release of that. So again, while SPR sensorgram, KD value can give you some number, but it's also important to look at what is the K small a on rate and K small d off rate. So that actually helps you to also know the various nature of the, different type of properties. And therefore, not only the dissociation constant, but also the values for the on rate and off rate are equally important in looking at SPR-based binding. So those are some of the things which industry looks at very heavily when they want to characterize their molecule. Since they're giving you the flavor of the technologies, let's now move on to one more platform, which is known as the octet-based platform, which is the BLI, biolayer interferometry technology. And unlike the SPR, like as you have seen SPR, we are immobilizing your protein on the chip, and we have 4 channels. So theoretically, we can do 4 protein type of interaction or 4 different concentration, which we can do the binding and look at their interaction. But another approach, which is based on the interference-based, looking at the changes, it is based on the solution-based form, which is one of the very different and added advantage to look at the protein-protein interaction. Here we are looking at directly the various type of biological samples in the solution form and trying to measure that their binding in the solution form itself. So think about the clinical samples. And if these clinical samples are going to be utilized again for some application, then the BLI can be really, a strong approach, because you're now going to, do the dipping and do the interaction with the BLI and use the same sample now for further protein extraction, peptide preparation for doing the mass spec-based approach. The same sample can be utilized for multiple application as well. So BLI offers a couple of these advantages. Additionally, it can use the 96 well plate format, so much more high throughput screening can be done, unlike the 4-plex which you have seen in the SPR. So now let's go to the lab, and I request Nirshar to give you demonstration for BLI technology.
Hello. So first I'll show you. The instrument, how it looks like. Then we will look into the software, how all the things work inside the software. Okay. So as you might have seen somewhere, this is how a BLI looks like. You can see essentially there is a black screen so that no light comes from outside inside to this instrument, because this is a very light dependent and light sensitive instrument. Now there are 2 trays, one for where the analyte comes in, one where the sensor comes in. So let's get a look at the sensor up close. So this is this tiny compared to our fingers, and this is this slim. Here is the bio surface, where different kind of surfaces, be that streptavidin, be that protein ProA, or any other kind of tag can be done here, and the anti-tag will be in your ligand. So this is the robotic arm. It moves this way and completely to left or right. It takes the optical sensors from here and then different samples on this analyte chamber. Also, when we are using, we also need to have plates over here so that the sensors don't go dry. Now you can essentially see that the plates are black. which is again an indicative of no light reflection or no light coming into the plate. So everything, the only light source will be from the machine, which actually makes the difference of finding. Let's share the screen. I'll share the screen here. Interesting for me. Interesting for me. Not for me. Just a minute. So first, as you can see that here I have opened two experimental methods. So we'll have to define the plates where the ligands are there, where the buffers are there, and the analytes are there. The one plate that you saw on the right. And then the assays will be defined with steps like the baseline step, which how many seconds and how much the shaking speed. We generally shake it for 1000. And then these are the seconds that we are talking about. Then the loading, then the baseline, and also the positions where we are talking about. So see this sample one. So one is here, then two, then three, like that, with step where it is going. Also, we need to show the sensor assignment where on which of the positions the sensors are there. So we'll have to make sure that it is on a single line because it cannot take randomly from here. Throughout the line, we can go up to 8, or we can go obviously 1 or 2. And then we have this beautiful step of doing the review of how the experiment will look like. So if any step we missed out, we can cross check, and then obviously we can run. So this what on the left we have shown is the kinetics. On the right, we can also do quantification, which is a much simpler, where we have different concentrations and just like that. So this is a much simpler, similar kind of setup. Let me show how the results looks like. So this is how when we open, this is the kinetics experiment that I was showing. So these are the different concentrations of the analyte which has bound. The first height that you are seeing is the loading, where the ligand loads onto the sensor, be that biotin coated protein that binds to the streptavidin of the sensor, or so on and so forth, different kind of chemistries. One part of the chemistry has to be bound to your first ligand, and the second part is obviously already bound to the sensor. Like streptavidin is already bound to the sensor, and we have to biotinylate our proteins so that that interaction happens. So loading, then baseline, then association, dissociation, like that it goes on. So one step you might have we might have missed in saying there's a regeneration after every association dissociation where the analyte gets dissociated completely from the ligand so that the new association can happen from almost base level 0. So that's what that regeneration is about. It doesn't take off the ligand from the sensor, but it definitely takes off the analytes which are bound. So we will go for the pre-processing, where first we need to, just like we do controls in our experiment, one sensor has to be without the ligand so that the data that comes out that will be subtracted from the one that has the ligand. So similarly, blank subtraction kind of stuff. Same for the reference sample. One sample will be there that won't have the analyte, only the buffer. That will also get subtracted. And then the data correction, we can take baseline step, association, or something like that, and another interstep correction, so that all of these looks like this, one common start and common end, like that from the association dissociation step as well. Then we go, obviously we do some kind of filtering, and then we go to the kinetic analysis, where we can choose to decide either by the basis of association or dissociation or both. Here we also have model if we know that it is one molecule versus one molecule they are going to bind, or it's there is a dichotomous like 2:1 where the ligand is higher, 1:2 where the analyte is in more number, we are expecting it to bind, like that. Also, if we have different compounds, we can go for local, that each and every of these graphs are of different analytes, or if it is the same analyte, different concentration, then we'll have to do the global. So like that, we get into the analysis, where we get the Ka, Kd, and finally the K capital D, the rate constant, where when we are doing it global, we can see it's the same rate for all the concentrations that we have seen. And also, people doing enzyme kinetics can also do the steady state. They'll also find the KVM, which is also again the same as calculated from here as well. So this is how the things looks like. Any kind of interaction studies can be done in this instrument. If we have just 1 second, I'll share one more thing. So what sir has already told. So basically the difference is the light that reflects back. That's where the difference happens. When there is nothing bound, there will be a reflection pattern. When there will be the first ligand bound, there will be a shift in the reflection wavelength. Then when the analyte bound, there will be further shift. So this shift, this delta of the lambda is what we calculate by how much it is binding, by saying how much it is binding. That is the wavelength shift we are calculating. So, and these are how experiments are set up. Just a second, I don't know why it is not going. Let me just straight ahead. Let me just straight ahead go to this. So these are the various type of biosensors that they provide. Even more, there are different kind of chemistries. One would be bound to the ligand. One would be bound to the sensor. The other you'll have to pair it up with the ligand, bind it up with the ligand. We commonly use the streptavidin sensors, and sometimes we use the pro sensors as well, just basic protein quantification. So yeah, these kind of things we do, and also various kind of experiments can be done based on any kind of one versus one interaction. If we are trying to do that, we can do in this software and in this technology. So to conclude everything, this instrument, just like SPR, can also do different kind of interaction studies, be that antibody, antigen, be that drug, protein, or any kind of molecule studies that can be done. One can be bound to the ligand, one can be the analyte. So that's how versatile this instrument and technology is. So now level 3, obviously, as Prof said. Now back to Prof. Ansha.
All right. Thank you, Rishir. So this was a good demo for the BLI, and what a student would have noticed that the concept remains same. You're looking at the biomolecular interaction. You're utilizing different type of platforms. We showed to you protein microarrays, peptide microarrays. Now we showed you the SPR and the BLI. Of course, this is not the end of it. There are many other platforms available. We just chose it because of availability in our lab and most commonly utilized platform for high throughput protein-protein interaction. But now let's say you generate a lot of these data and you want to really look at much more systems level information, because if you think about biology, we are looking at how all these perturbations changing towards addressing some of the physiological questions or some of the very key biological questions which you are very interested to learn. So how to take now this type of data forward and analyze that data. So now Arga is going to give you a demonstration for a few softwares to look at how to plot different type of protein network analysis and make good displays of your data. So over to you, Arga, for demonstrating some softwares for looking at the protein interaction analysis.
So hello everyone, welcome to the few of the software. The two commonly available softwares which are known online are String and Reactome. So I will start with the start. So you can start by searching the String database. So this is the String database. So I will just go to the home. So this is the String database. Here you can put your significant proteins, which came to be very much significant. So I have put few of the human proteins which were differentially regulated. So String database basically gives you the interactions. Okay, basically the interactions what are there, like which all things are there. So I will just go for search. Maybe you can also Yeah, I can click on the type on the name as Homo sapiens. Wherever you are comfortable, you can put in. So yeah, I got the Homo sapiens. Then I go to continue. And 31 based on the 31 input proteins or the autoantibodies or whatever proteins you are significant, it will tell you like it has found a match with the human database or not. When I click on continue, below is the details of it, like, so this is the string interaction. Below you can find a lot more. Again going to for Homo sapiens. So this is the pattern what you will find after finding the interactions, like you can interactions you can find for the legends, viewers, settings. So in the settings you can select a full string network, the protein association. This confidence, like how is the confidence of the interaction? I want to go with the highest confidence, which is 0.9. You click on that. Maximum number of shells you can put to 350, 10 interactors. I can just looking at a sub particle, I can include 10 interactors, and I want an interactive PNG. Once this is being done, I will click on the update. So you can find so these are the, so you can find like many of the interacting proteins will be there which are not mapped, but few will be mapped. These are your high in confidence proteins which have strong interaction, and also it is backed up by the evidences. In the analysis, like you can find, it gives a brief analysis. The biological, if you go for the gene ontology study, basically whatever proteins I have indicated, 13 out of it matched for the acting cytoskeleton organization, like out of that, which is very high. So you can clean or click on each proteins, each of the bar biological function, you can find the geoterms. You can click on it and you can come to the geoterms, like which will we go. So amigo 2 is linked with this screen. So each protein, like the accession, there are annotations, like in each organism, their graph views, how they look like, their info tree, their neighborhoods, their mappings, everything will be detailed in that. So once this is done, like once you are confident on the analysis, like which pathways, which means which interactors you are selecting, like I have the biological process, I have the molecular function, I have the cellular components on the basis of which can be selected. Once you are very much confident enough, which you are taking, you can just go to the clustering option. You can perform the k-means clustering, obsolete as well as the k-means. So I generally go for the k-means to identify the accurate clusters which are happening, like the high confident interactors which are coming in the study. And once we do the k-means clustering,
adjusting the k value parameters. So you can find the main clusters which can be depicted. So this is my one of the main clusters. So here it is written cluster 1, which comes for the platelet aggregation. 14 proteins, like the 14 genes, have been counted for the clustering for high component cluster, which is mapped to only platelet aggregation. Complement activation, 5 of them have matched. Spectrin associated cytoskeleton have been matched in that. So once that this is being done, I am going to show you one more software which is called Reactome. I can select my protein list which is present. I can select my protein list. I can come to the Reactome. So this is how a Reactome looks like. I can click on the search site. Just I will go through the start. Come to Google, search on Reactome, click on Reactome. You will be launched into the Reactome Power database. You go inside the Reactome. It is a bit slow, like you require a high interface net for Hello. I think Reactome is down now, but you can find various tutorials which are also available online for the Reactomes. I think the site is down now. Yeah, it's open. I think the pathway browser is currently not working properly. Prof, we can explain this software sometimes else.
Yeah, we can also show maybe, a string or something, some other network analysis software in the meantime. But idea is that,
Yeah, string I have
how to make meaningful sense of your various type of changes which you see, because while you will see expression of many proteins are changing, but until unless you narrow down to which pathways many of these changes belong to, you cannot think about the physiological consequence of it, right? So therefore, some of these softwares are quite helpful. But given the timing is getting short now, I think, Arget, will be a good idea if you can now show them some detail about the mass spectrometry based approaches. So let's have a quick preview of different type of mass spec which we have available. And then we will talk briefly about how mass spectrometry is utilized for different interaction analysis. So Arget, please go to the, the MS facility and let us first show the different instruments to utilize for the MS-based interactomics.
So this is the orbital fusion, what you are seeing. So this is a hybrid mass spec. It has a robust capacity of doing both global as well as the targeted proteomics. This is a nano LC. You can find this is a nano LC because it is specialized for only proteomics workflow. You can place your samples inside the sample tray into the nano LC. Once the sample has been injected, like once the sample is being placed, it has the quaternary pump. The samples are eluted coming from the nano LC to the trap column where all the residual elutes are filtered. Then actual loading happens in the main column, which is the C18 PepSip map column, and finally it enters into the isothermal capillary or the high voltage region. From the high voltage region, it then means travels to the through a series of quadrupole, it travels to the C trap where the ions are trapped, and finally moving to the orbit trap orbital. So this is how a chromatogram looks like. Like you can find this is a 120-minute gradient. This is a 120-minute gradient. Each MSMS peak corresponds for more than 2 MSMS. And here you can do both targeted as well as means global proteomics, and you can find your best interactors which are inside. This is one more instrument which is Orbitrap Q Exactive. This is an ultimate 3000 HPLC. The samples can be placed in the samples compartment. We can generally use for the small molecule interactors as well as the metabolite interactors or protein metabolite interaction studies. So the column is placed in the column compartments. The sample is loaded. So this is the column compartment. I think maybe sometime later I can show you this. Yeah, so the samples travel from the column compartment to the main mass spec. From the main mass spec, this is the hazy probe. What you can see from the hazy probe, the samples are injected, and you can see the sample is currently running now. A live acquisition is happening, and this is how the peaks looks like. Like this is how the peaks looks like for the data acquisition. One more mass specs can be used, which are the triple quadrupole series. These mass specs are generally used for the validation of the interactors or the bait or the interaction. So this is a thermo-vanquish UHPLC. The samples can be loaded into the sample compartment here, like you can find. So this is the sample compartment region. Once the sample has been loaded from the quaternary pump, it comes to the mass spectrometry, like the mass spectrometry. The quadrupole is available. The samples are injected, and you can find the peaks like if I can show you one file. So this is how the single interactor peaks of the proteins or the peptides look like. So this is one of the interactions found for the tetrahydrochlorate or the chlorine, like and also you can find what all proteins are available. So I think there is one more mass spec that Dr. Sambhu is going to introduce. Dr. Sambhu, you can be fast and crisp.
In addition to various type of Orbitraps, which you have just seen, we also have a 4D omic technology, which is the timsTOF. So Shambhu, please share your video now. I'll add in spotlight. Yeah. Please show the timsTOF and talk about it.
So I will briefly introduce the timsTOF Pro 2, a mass spectrometry designed for the high sensitive and high throughput proteomic study. It integrates the TIMS, which is trapped ion mobility.
Shambhu, you have only 1 minute time, so please just quickly show the instrument. I think that's all. You just show the instrument, I can explain. All right. So,
this is the timsTOF.
No, Shambhu, please give me time. So what you see is another mass spec, which is the giving you some more spatial information with the CCS value, cross-sectional information which we obtain, and that is the giving us the 4D separation with the timsTOF technology. Along with that, we also have a very robust chromatography, which is the EvoSep chromatography interface with that. So I think I'll stop here for the mass specs, and I'll quickly just tell you that while we have talked to you many type of array and many type of label-free biosensors, in addition to that, there are many mass spectrometry-based approaches which are coming forward, and those are quite important for us to start knowing about it and start thinking how to utilize your interactomic analysis using mass spec. For example, affinity purification-based mass spectrometry, APMS, many type of proximity labeling-based approaches are also quite important and getting more and more interest and attention. Various type of cross-linking mass spectrometry-based approaches have also come forward to look at much more structural interactomic-based approaches. So now people are trying to integrate different platforms because one platform cannot give us all the information. If you can think about some innovative ways of labeling your proteome, mass spectrometry can be very strong and good way of looking at the interactions. But if you have to look at from the complex samples and especially biological samples like blood and plasma and serum, then I think the protein microarray platform could be much more powerful. If you're looking at their accurate quantification and accurate KD values, then I think label-free biosensors like SPR or VLI will be very helpful. So let us stop here. I hope you got the good flavor and the recap of various technology platforms which are used for interactomic analysis. Let me thank various TAs, Dr. Argya, Dr. Shambhu, Nirjar, Vaishali, and many people from the lab who have been involved in giving you demonstrations. I hope you learned a lot of new things from this course, and soon you will be writing the examination for the MOOC course on Interactomics. If you happen to be one of the toppers of the course, we'll be happy to get in touch with you to give you opportunity to participate in our upcoming international conference, which is APT, Advances in Proteomic Technologies, which we are going to conduct at IIT Bombay from June 27 to 29. We'll be happy to give you the free registration opportunity to come and attend this conference. We wish you good luck, and if there are more queries, we'll be happy to talk to you.